Hello, welcome to AQA demola, and today's lesson we're going to be covering protein synthesis. We're going to go through it all: transcription, translation, RNA, even thymine to uracil. Let's explain it. Okay, no faffing about, we're going to get straight into the process of transcription. So, number one, this happens in the nucleus of our cells. So, I don't think we've actually got to cell structure yet, but when we do, we're going to cover it. Transcription occurs in the nucleus. So, step one, DNA helicase. This is our guy, the scissors, yeah? It's going to break hydrogen bonds between your nitrogenous bases on separate strands. So remember we have adenine to thymine, cytosine to guanine, yeah? It's going to break those bases, as you've shown with the big X there. DNA helicase, now, specifically, when we're talking about transcription, it causes DNA to unwind at a gene. So remember this phrase, at a gene. You need to be very specific when it comes to transcription. So instead of the whole strand unwinding, it's only going to be a part, a gene, that unwinds. Now what's next? So we're going to have a sense strand and an anti-sense strand. Your sense strand runs from 5' to 3'. Cool. So that's going to be your template strand. And the free-floating complementary RNA nucleotides are going to line up with the exposed bases on that sense strand. Yeah? So you can see that diagram here, sense strand 5' to 3'. You can see in orange, the complementary free-floating RNA nucleotides are lining up with that one, whereas our anti-sense strand, this guy down here, anti-sense, he's just being left alone. He's not involved. Cool. Just know, very important point. Say this every single time. In fact, we're gonna put ticks next to it. This will get you a mark every single time in an exam. Thymine is replaced by uracil. Very easy mark to pick up whenever you're talking about DNA. When you're talking about transcription, just say thymine is replaced by uracil. So now your competency-based pairings are adenine to uracil, cytosine to guanine. Yeah? Cool. What's going to happen next is RNA polymerase. So remember, before it was DNA polymerase, now it is RNA polymerase. So it's going to catalyze the formation of phosphodiester bonds between adjacent RNA nucleotides and condensation reactions. Again, very, very similar to what we were doing in DNA replication. So the RNA polymerase, phosphodiester bond here, there, all across the chain, and you create what is called a strand of mRNA, which we'll highlight there, a strand of mRNA. Yeah? Now that strand of mRNA is going to detach from the nucleus, and it's going to detach from the DNA. Yeah, it detaches, and then it leaves the nucleus through the nuclear pore. So again, we're going to get to this more when we talk about the cell structure, but these gaps are called nuclear pores, and that mRNA is going to just wiggle out of that. Another important point to mention is that DNA is too big. So DNA, too big to leave. So that's why we need to go through this process of transcription so that we can get a small enough, not really a copy, but a small enough part of the DNA out so that we can make our proteins. And then DNA is just going to rewind to form the double helix. Guys, I promise you, go through that. If you are able to kind of regurgitate all of that in an exam, you'll be scoring basically all of your marks when it comes to explaining the process of transcription. Okay, so we're going to keep pressing on part 2, translation. So if you remember back to your cell structure, that's your cell, this is your nucleus, and the ribosomes are all kind of dotted around here. This is where translation is going to occur. So your mRNA exits the nucleus through your nuclear pore, and then it finds itself in one of these ribosomes, and now we're going to focus on, yeah? So your ribosome is made up of 2 subunits, your 60S and your 40S, and that is all in an 80S ribosome. We will get into this later in the topic, but guys, I've not made a mistake there. 60 and 40 does not add up to 80, but those are your subunits, yeah? And what's going to really happen is the ribosome is going to be moving along that strand of mRNA, yeah? So you can see the codons here. A group of 3 consecutive bases is a codon, as you should know from GCSE. But literally, it's going to be, so from AUG, moves to CAG, moves to CCC, moves to AGC, yeah? So it just keeps coming along, and it begins with our start codon, yeah? Now, every single time a codon is read, it signals for a tRNA molecule that's complementary anticodons. So like, for example, AUG, if we're talking about the complementary anticodon to that, we would be saying UAC. Get what I mean? So each tRNA molecule has those anticodons, as I've tried to draw here. Those are the anticodons. And they line up with the specific codon, and they each carry a specific amino acid. So you can see at the top here, if I just highlight them in yellow here, this is an amino acid, and every single tRNA molecule carries one of them. So as the ribosome is moving along the chain of mRNA, you're getting a tRNA molecule with a specific amino acid, which is going to end up making a specific protein. Now, because we're so close up together, what's going to happen is peptide bonds are going to form between adjacent amino acids. So just because they're next to each other, peptide bonds will form. I just thought it might be nice to quickly show you what a peptide bond looks like. So if we draw out an amino acid, like so, what you're actually going to see is going to be double bond O, and then N, H, and then so on and so forth. We're literally just going to copy it again. So like, I should have just done it like this. And now you can see you've got a peptide bond in there, which will highlight in yellow. That's a peptide bond, yeah? Keep it common. So if you know your chemistry, that will be an amide. Yep, cool. So once the stop codon is read, so we keep going along that strand of mRNA until we reach a stop codon. And when that's read, that's a signal for the ribosome, okay, you're done here. I'm going to leave. That leaves your sequence of amino acids, your chain of amino acids joined by peptide bonds. That is a primary structure protein. Protein structure will be in a later video. That is our process of translation. So I hope that made sense as well. Again, if you need to rewatch it, try and really visualize it, break it down. That is what's happening in translation. I just thought couldn't end this video off without talking about our genetic code. So, what is a gene? Guys, perfect definition for a gene is a sequence of bases in DNA coding for a specific protein or polypeptide, whatever you want to say. That is a gene. And each gene has its own unique sequence of DNA bases to code for a specific protein. Yeah? That's what you kind of need to know about genes. And I just thought there are kind of three main things that you need to know about codons. Yeah? So we said that a codon is a sequence of 3 consecutive bases that code for a specific amino acid. That was our definition. So you need to know non-overlapping. So they're all coding for a specific amino acid, which I feel like is a kind of awful definition to describe what non-overlapping is. But that's the one that you would say in an exam. I'll show it here. So you can see here, if we look at the bottom, the non-overlapping code, which is what it is, you can see that we've got number 1, you've got 3 bases, number 1, then you move on to the next 3, number 2, then you move on to the next 3, number 3. Whereas at the top, if we had overlapping code, which we don't have, what you would see instead is that you can see there's the first codon here, 1, 2, 3, but then we break it up there for the second codon. We use part of this to go for our next 3, and we're even breaking something there to go for the next 3. That gets way too complicated. We do not do that in organisms. I hope that makes sense. Then degenerate. So some codons code for the same amino acid. Let's quickly show an example here. If I can quickly spot one, I have spotted one. So you see leucine here, UAG. So you could be UUA. You need to learn how to use these diagrams, by the way. You start with the big one, then you go to this one, then you go to one of these. So if we're talking about leucine, it would be U, and then A, or UUG. Yeah, that's leucine. We can also get leucine by C, U, and then whatever one. Yeah, just be aware, some of them will code for the same. And I think, yeah, it's shown a lot more clearly here, like all of these are serine, for example. All of these are leucine, for example. You just kind of need to be aware that some codons will code for the same amino acid. Yeah? Another thing, genetic code is universal. So every single organism is going to use as bases adenine, cytosine, guanine, uracil, thymine, whatever. Every single organism is going to use those bases. So that's another thing to be aware of when we come to genetic code. Okay, so it's question time. We've got 3 here. First question, comparing and contrasting transcription and DNA replication. Guys, this question is so common, so be prepared to answer it. The next one, definition of a gene, and then the role of tRNA in the production of leptin. So I'm going to give you guys a moment to try those questions. Okay, we're really going to focus on this comparison question, yeah? Guys, I want you every single time to see this question and start smiling. Cool. That's what we want to get for. So, similarities between DNA replication and transcription. Well, we said that both of them involve DNA helicase at the very start to break those hydrogen bonds. Both of them involve the formation of, honestly, I would go phosphodiester. Very easy to remember. So phosphodiester bonds forming between DNA nucleotides, if we're talking about DNA replication, and forming between RNA nucleotides, if we're talking about translation. Those are some easy similarities. They even said that they will allow you to say condensation reactions occur. Guys, literally any of those, they will get you a mark. So just be ready for that. Differences are a bit easier to do, yeah? One of the easiest differences to say, RNA nucleotides versus DNA nucleotides, yeah? RNA polymerase, DNA polymerase, 2 easy ones there. And then we can say honestly, this point about transcription produces a single strand of mRNA. Fine. DNA replication, double stranded. So DNA replication, double stranded. I would go single strand, double strand. Very easy to do. Yeah. So, guys, if you need to remember those points, similarities, you're saying phosphoester bond formation and DNA helicase. Differences, DNA nucleotide, RNA nucleotide, DNA polymerase, RNA polymerase, and single strand of mRNA versus double stranded DNA. That's the products. Those ones should be fine. I don't know why this is in the allow column, but adenine with uracil instead of adenine with thymine should also be one that you can get. Yeah? Cool. Then when we're talking about defining a gene, literally almost like what we said, you need two parts to this definition. So your first part is going to be a sequence of bases in DNA. Guys, very important. In DNA. That one, if you don't say it, you don't get the mark. Yeah? Very, very strict, I know. And then your second part is going to be codes for, I would normally say a polypeptide, but like it shows, so you can say amino acid, you can say a primary structure protein. Literally doesn't really matter. Codes for protein. Yeah? Cool. And then when we're talking about the role of tRNA in the production of leptin, that's kind of, it's a trick question in a way. Leptin's a protein, so it just wants the role of tRNA in protein synthesis, yeah? So literally, exactly what we talked about when we said translation. tRNA molecule, it brings in an amino acid. So the bringing the amino acid, we said it has anticodons, and then we also said it carries a specific amino acid. So just be aware of those kind of things when you're answering questions like. Not gonna lie, it was a rough day at the office thinking about the ASR tips, but we got a few for you. Be very comfortable comparing DNA replication and transcription. Guys, it's like, just because of how common that question is, I would have an answer literally just ready to go off the bat as soon as you enter the example, just in case. Features of genetic code, those three things, non-overlapping, degenerate, universal. If it comes up, it's marks that you really don't want to miss out on. So I just remember those three in your head. We've also got the genetic wheel. I did show you when I was talking about degenerate or genetic wheel. Those ones can be difficult to navigate. So I would definitely practice in your own time. Guys, everyone's using ChatGPT these days. So if you need to pop one image into ChatGPT and then practice with ChatGPT, like asking it, identify which codon codes for lysine, leucine, whatever amino acid you want. If you need to do that, it will help you. Because if that comes in an exam, you don't want to be confused and not get what isn't the most difficult mark. Yeah? What should we not do? Enzymes. So I will always emphasize this. DNA polymerase, RNA polymerase catches out somebody every year. Don't let it be you. DNA replication transcription, again, it happens to someone every year. Don't let it be you. And thymine replaced by uracil happens every year. Don't let it be you. So those are just three common errors, but they're not errors that you want to be making. Yeah, just remember those. Cool. That is a wrap for this video. So thank you all for watching so much. I hope it's been helpful. Literally, any questions about any of this, drop it in the comments below. I'll do my best to answer. We got the TikTok and the Instagram, both at demola. But if it's been helpful, like and subscribe, and I'll catch you in the next one. Bye for now.